The aim of the presentation is to give an introduction to the regulatory requirements of developmental and reproductive toxicity testing for human pharmaceuticals, as harmonized on the ICH level. Before ICH was established, efforts on a harmonized testing approach were already undertaken, aiming consensus for regional differences in the general approaches and experimental details. The parent guideline goes back to 1993, with an addendum on toxicity to male fertility in 1995. My name is Günther Wachsnecker. I am from the Austrian Medicines Agency and acting on behalf of the European Commission as rapporteur for the third revision of this guideline. The third revision of the guideline reached step 4 of the ICH process in February 2020. It incorporates experience gained with the testing of pharmaceuticals using the current and novel testing paradigms, as well as the advances of scientific, technological and regulatory knowledge over the past years. This revision intends to provide greater clarity and alignment with other ICH guidelines, including ICH M3, ICH S6 as well as ICH S9. Usually, even after marketing authorization, clinical experience about the risk of toxicity to reproduction is limited. Consequently, this risk remains a key non-clinical safety aspect, starting with the initiation of clinical trials up to the marketing phase. The guideline aims to reveal any effect of the pharmaceutical on mammalian reproduction and development relevant for human risk assessment. It is a key principle that the risks to all stages of reproduction and development should be assessed, unless the stage is not relevant to the intended population. Key principles introduced with the third revision are: to bring the guideline into alignment with other ICH guidelines. It elaborates on the use of exposure margins in dose level selection. It incorporates a section on risk assessment and expands the scope to include vaccines and biopharmaceuticals. It gives a rough description on the qualification and use of alternative assays for developmental toxicity testing. The guideline contains 2 annexes. Annex 1 addresses the design and conduct of in vivo reproductive toxicity studies, while Annex 2 outlines points to consider in the development of in vitro, ex vivo and non-mammalian in vivo assays, their qualification and potential use to support reproductive safety assessments. During expert working group discussions, it was recognized that further details complementing both annexes will be needed in the future to achieve the intended goals of the revised guideline in a harmonized fashion. Thus, a maintenance procedure is applied to both annexes. The objectives of the guideline are to provide recommendations on international standards for the assessment of non-clinical BARD testing, supporting clinical trials and marketing authorization, and the description of potential strategies and study designs to supplement available data to identify, assess and convey risk. The guideline applies to pharmaceuticals including biotechnology-derived products, vaccines and the novel constitutive ingredients for infectious diseases, novel excipients. It does not apply to cellular therapies, gene therapies and tissue-engineered products. Whether and when non-clinical studies are warranted is determined by ICH M3, S6 and S9. The content is separated into following sections: general considerations on reproductive toxicity assessment, design and evaluation of in vivo mammalian studies, test system selection, dose level selection, route of administration and schedule, possible combination study designs in rodents, data reporting and statistics, principles of risk assessment. The following stages of reproduction should be assessed: if relevant to their intended population. A. From premating to conception. B. From conception to implantation. C. From implantation to closure of the hard palate. D. From closure of the hard palate to the end of pregnancy. E. From birth to weaning. F. From weaning to sexual maturity. The timing of studies is dependent on study populations and phase of pharmaceutical development as outlined in the respective ICH guidelines. Typically, 3 in vivo study types have been evaluated: a fertility and early embryonic development study, FEED, stages A and B, embryo-fetal development studies in 2 species, EFD, stages C and D, and a pre- and postnatal development study, PPND, stages C through F. The stages covered in individual studies are left to the discretion of the sponsor, although the timing of studies within the pharmaceutical development process is dependent on study populations and phase of pharmaceutical development. Following key factors need to be included when an overall integrated testing strategy is developed: the targeted patient population and conditions of use, especially in relation to reproductive potential and severity of disease, the formulation of the pharmaceutical and routes of administration intended for humans, relevant data on toxicity, which can also include data from in vitro, ex vivo and non-mammalian studies and structure-activity relationships, but also pharmacodynamics, pharmacokinetics and pharmacological similarity to other pharmaceuticals, and aspects of the general biology of the pharmaceutical target or known roles of the target in reproduction or development. The target patient population and therapeutic indication can influence the extent of DART testing. Studies evaluating all stages of reproduction and development are not warranted if the disease indicates that developmental and reproductive toxicity will have minimal impact on the risk of the pharmaceutical in the target population. For example, studies covering all stages are not necessarily appropriate for an exclusively postmenopausal female patient population, for use in the pediatric or juvenile prepubescent population, or for patient populations in hospitalized settings where pregnancy can be excluded. Before designing a testing strategy, it should be determined if the intended pharmacologic effects of a pharmaceutical are known to be incompatible with fertility, normal EFD, or assessment of particular endpoints, e.g. general anesthetic and assessment of mating behavior. This assessment can be based on data with other pharmaceuticals with similar pharmacology, known effects of target engagement, or on knowledge of effects in humans with related genetic diseases. For example, it would be appropriate to modify the design of a PPND study for a pharmaceutical developed to prevent preterm labor, if the intended pharmacologic effects are incompatible with the study endpoints. Testing for a particular reproductive endpoint is not warranted with justification. Toxicity considerations. Repeated dose toxicity studies with sexually mature animals can provide important information on toxicity to reproductive organs that can affect the design of a DART study. The existing toxicology data for the compound should always be considered, taking into account the dose levels, toxicokinetic profile and dosing duration. For example, the standard fertility study design can be modified to alter the duration of dosing or the start of cohabitation for a compound that affects testicular tissue. Timing considerations. General guidance on timing for conduct of studies assessing reproductive and developmental endpoints is described in ICH M3, S6 and S9. The timing for when to conduct specific DART assessment should take into consideration the need for these data to support the safe use of the pharmaceutical in the clinical trials or the intended patient population. Consequently, it can be appropriate to consider altering the timing of the assessment of specific reproductive stages. In addition, under limited circumstances, results of properly qualified alternative assays, in vitro, ex vivo and non-mammalian in vivo assays, have the potential to defer or replace conventional in vivo studies. Toxicokinetics. Exposure data can be generated in either reproductive or repeated dose toxicity studies. However, given the potential for meaningful changes in TK parameters induced by pregnancy, it is recommended to determine if pregnancy alters exposure. If dose selection is based on exposure ratio, GLP-compliant TK data in pregnant animals is expected, and the sampling days should be justified. When warranted, determination of the pharmaceutical's concentration in the embryo or fetus can facilitate interpretation of discordant or equivocal evidence of developmental hazard. This information can be collected in a separate study to determine the actual exposure. However, a direct comparison to the potential levels in the human conceptors is not appropriate. Evidence of lactational excretion can be obtained when warranted by sampling milk or by demonstrating exposure in offspring during the pre-weaning period. General concepts regarding TK data collection are discussed in ICH S3A. The design and evaluation of in vivo mammalian studies. The strategy to evaluate the potential reproductive and developmental risk of a pharmaceutical generally includes one or more in vivo studies. The key factor is that in total they leave no gaps between stages and allow for evaluation of all stages of the reproductive process, although in some species, e.g. the non-human primate, it is not possible to evaluate all stages. For most pharmaceuticals, the 3-study design will usually be appropriate, although various combinations of these study designs can be conducted to address specific product needs and to reduce animal use. Study details for the FEED, EFD and PP&D studies, and combinations thereof, can be found in Annex 1. The stages covered in individual studies are left to the discretion of the sponsor. The aim of the fertility and early embryonic development study is to test for adverse effects resulting from treatment initiated prior to mating of males and or females and continued through mating and implantation. This comprises evaluation of stages A and B of the reproductive process. Results from repeated dose toxicity studies of at least 2 weeks duration can often be used to design the fertility study without conducting further dose-ranging studies, although studies of such short duration can be insufficient to reveal all adverse effects. A mating phase is expected in most cases when a FEED study is warranted to support exposure of the target population. Such studies are typically performed in rodents. If no adverse effects on fertility are anticipated, both sexes can be treated and cohabited together in the same study. If effects on fertility are identified in the study, the affected sex should then be determined. In contrast, if adverse effects are anticipated based on mode of action or on the results of repeated dose toxicity studies, each treated sex can be cohabited with untreated animals of the opposite sex. This can be achieved using separate treatment arms within a single study or by conduct of 2 separate FEED studies. Reversibility of adverse effects on fertility and early embryonic development can have an important impact on risk assessment. The FEED study design in female rodents allows for the detection of effects on the estrus cycle, tubal transport, implantation, and development of pre-implantation stages of the embryo. When estrus menstrual cycles are evaluated, it is important to obtain baseline cycle data over 2 or 3 cycles minimum to distinguish between treatment-related effects and intra-animal variability. The monitoring of estrus cyclicity should continue through the time of confirmation of mating. The FEED study design for male rodents that includes 2 to 4 weeks of treatment prior to cohabitation allows for the detection of effects on spermatogenesis and epididymal transport. When data from repeated dose studies suggest toxicity to the testes, it can be appropriate to extend the duration of pre-cohabitation treatment to 10 weeks. This permits assessment of effects on the full spermatogenic cycle as well as epididymal transport. The FEED study additionally permits detection of functional effects, e.g. on libido, epididymal sperm maturation, ejaculation, that cannot be detected by histological examinations of the male reproductive organs. When there is cause for concern based on mode of action or data from previous studies, additional examinations can be included in repeated dose toxicity and/or fertility studies, e.g. sperm collection for counts and morphology, motility assessments, measuring hormone levels, or monitoring of the estrus menstrual cycle to further characterize potential effects on fertility. If the biopharmaceutical is pharmacologically active in rodents or rabbits, a FEED study in one of these species is recommended. Mating evaluations are not generally feasible in non-rodents such as dogs and non-human primates. For example, if NHPs are the only pharmacologically relevant species, as for many monoclonal antibodies, see ICHS6, histopathological examinations of the reproductive tissues from the repeated dose toxicity studies of at least 3 months duration can serve as a substitute for the fertility assessments. Such an approach should include a comprehensive histopathological examination of the reproductive organs from both male and female animals. Unless the biopharmaceutical is intended to treat advanced cancer, in which case FEED studies are not warranted, animals should be sexually mature at study initiation in order for an adequate evaluation of the reproductive tissues to be made. These data would only provide information on the structure of the reproductive tissues, as no functional assessments of fertility can be made, and predicting effects on fertility and early embryonic development is not always possible, based solely on the results of histopathology assessments. The aim of the EFD studies is to detect adverse effects on the pregnant female and development of the embryo and fetus following treatment at stage C of the pregnant female during organogenesis. EFD studies include evaluation of fetal development and survival, stages C through D. For most small molecules, effects on EFD are typically evaluated in 2 species, rodent and non-rodent. At least one of the test species should exhibit the desired pharmacodynamic response. If the pharmaceutical is not pharmacodynamically active in any routinely used species, then routine species, genetically modified animals, or use of a species-specific surrogate molecule, e.g. in the case of oligonucleotides, can be considered, provided there is sufficient characterization of the model to ensure pharmacologic relevance. Genetically modified animals and surrogate molecules are generally most useful for hazard identification, but have limitations when used for risk assessment. Even when there are no relevant models, e.g. the pharmacological target only exists in humans, either normally or in the disease state, EFD studies should be conducted in 2 species to detect the adversity of off-target effects or secondary pharmacology. Clearly positive results for the induction of malformations or embryo-fetal lethality (MEFL) in a single species at exposures similar to that at the projected clinical exposure at the maximum recommended human dose (MRHD) can be sufficient for risk assessment. Under limited circumstances, other approaches can be used in place of definitive EFD studies. Alternatively, there can be adequate information to communicate risk without conducting EFD studies. Evidence suggesting an adverse effect of the intended pharmacological mechanism on EFD, e.g. mechanism of action, phenotypic data from genetically modified animals, can be sufficient to communicate risk. The effect of biopharmaceuticals on EFD should typically be assessed in 2 species, one rodent and one non-rodent, if both are pharmacologically relevant. However, the rodent is often not pharmacologically relevant, in which case EFD assessment in a single pharmacologically relevant non-rodent species can be conducted. In cases where the NHP is the only relevant species, an enhanced pre- and postnatal development study can be conducted instead of an EFD study. Biopharmaceuticals intended for the treatment of advanced cancer typically need only be assessed in a single pharmacologically relevant species. See ICH S9. When no relevant species can be identified because the biopharmaceutical does not interact with the autologous target in any species relevant to reproductive toxicity testing, use of surrogate molecules or transgenic models can be considered, as described in ICH S6. Calculating safety margins relative to human exposures with surrogate molecules is not appropriate. If there are no relevant species, genetically modified animals or surrogates available, in vivo reproductive toxicity testing is not meaningful. In this case, the approach used for risk assessment or rationale for not conducting studies should be justified. Alternative approaches for addressing EFD risk, the use of alternative assays. A number of alternative in vitro, ex vivo and non-mammalian in vivo assays have been developed to detect potential hazards to embryo-fetal development. They have been used as drug discovery screens for adverse effects on EFD and have assisted in the understanding of the mechanism of toxicity, which can be useful for translating non-clinical data to human risk, especially for human-specific targets. The continued use of alternative assays for these purposes is encouraged. If properly qualified, alternative assays have the potential to defer or replace, in certain circumstances, conventional in vivo studies. This has the added benefit of potentially reducing animal use. Concepts to consider when qualifying these assays, and examples when the use of such assays could be appropriate, appear in Annex 2. Approaches that incorporate alternative assays should provide a level of confidence for human safety assurance at least equivalent to that provided by the current testing paradigms described above. Based on the direction of scientific development as of the writing of this document, it is expected that for regulatory purposes multiple alternative assays will be used within a tiered or battery approach. These testing strategies will be qualified within a certain context of use, which is defined by the chemical applicability domain of the assay and by characterization of the biological mechanisms covered by the assay. Potential approaches to defer definitive in vivo testing as part of an integrated testing strategy. The design of an appropriate testing strategy relies on a cumulative weight of evidence approach. ICH M3 allows preliminary the toxicity data from 2 species to support the limited inclusion of women of childbearing potential, before conducting definitive EFD studies. Based on these considerations, this guideline expands on ICH M3 by allowing 2 additional options to support inclusion of WOCBP prior to phase 3 clinical trials. 1. Qualified alternative assays which predict the outcome in one species, see Annex 2, can be combined with a PEFD from a second species to enable the limited inclusion of WOCBP, up to 150 women of childbearing potential for up to 3 months. The alternative assay and the second species should generally cover both a rodent and a non-rodent species. 2. Additional endpoints incorporated into at least one GLP PEFD study, specifically increasing the group size of evaluable litters with inclusion of skeletal examinations, performed in a pharmacologically relevant species, if available, combined with a PEFD in a second species, allows all regions to include an unlimited number of WOCBP in clinical trials through phase 2. Strategy to address effects on pre- and postnatal development. The aim of the PPND study is to detect adverse effects following exposure of the maternal animal from implantation through weaning to evaluate effects on the pregnant or lactating female and development of the offspring. Since manifestations of effects induced during this period can be delayed, development of the offspring is monitored through sexual maturity. In essence, stages C to F. The rodent is usually used to assess PPND. However, other species can be used as appropriate. In most cases, a preliminary dose range finding PPND study is not warranted, because the appropriate information is generally available from prior studies. However, a preliminary PPND study with termination of the pups before or at weaning can be used to select dose levels or inform study design and/or to provide pup exposure data. If a modified PPND, ePPND study design is being considered to support pediatric development, see ICH S11. PPND considerations for biopharmaceuticals. For pharmaceuticals that can only be tested in the non-human primate, the ePPND study can provide a limited assessment of postnatal effects, but it is not generally feasible to follow the offspring through maturity. See Annex 1 and ICH S6. Test system selection, routine test species. Mammalian species should be used to detect DART. The use of the same species and strain as in already completed toxicity studies can eliminate the need to use additional animals, or conduct additional studies to characterize pharmacokinetics and metabolism, and/or for dose range finding. The species used should be well characterized and relevant for detecting effects on the endpoints in a particular study, e.g. with respect to health, fertility, fecundity, background rates of malformation and embryo-fetal death, etc. Selection of species for DART testing. The rat is generally appropriate for DART testing and is the most often used rodent species for reasons of practicality, general knowledge of pharmacology in this species, the extensive toxicologic data usually available for interpretation of non-clinical observations, and the large amount of historical background data. The mouse is also often used as the rodent species for many of the same reasons. For assessments of EFD only, a second mammalian non-rodent species is typically evaluated, although there are exceptions, e.g. vaccines and biopharmaceuticals. The rabbit has proven to be useful in identifying human teratogens that have not been detected in rodents and is routinely used as the non-rodent species based on the extensive historical background data availability of animals, and practicality. The animal species selected for testing of vaccines, with or without adjuvants, should demonstrate an immune response to the vaccine. The type of developmental toxicity study conducted and the choice of the animal model should be justified based on the immune response observed and the ability to administer an appropriate dose. Typically, rabbits, rats or mice are used in developmental toxicity studies for vaccines. Even though quantitative and qualitative differences can exist in the response, e.g. in humoral and cellular endpoints, between species, it is usually sufficient to conduct developmental toxicity studies in a single species. Although the degree and time course of transfer of maternal antibodies across the placenta varies between species, a developmental toxicity study in rabbits, rats or mice can still provide important information regarding potential embryo-fetal toxicity of the vaccine components, formulation, and safety of the product during pregnancy. Non-human primates should be used only if no other relevant animal species demonstrates an immune response. When there is a lack of an appropriate animal model, including NHP, an EFD toxicity study in rabbits, rats or mice can still provide important information regarding potential embryo-fetal toxicity of the vaccine components, formulation, and safety of the product during pregnancy. Non-routine test species. Species other than the rat, mouse or rabbit can be used to evaluate the effects of pharmaceuticals on various reproductive stages. When considering the use of other species, their advantages and disadvantages should be considered in relation to the pharmaceutical being tested, the study design and selected endpoints, and the ability to extrapolate results to the human situation. Non-human primates should be considered a non-routine species. They are most typically used for evaluating effects on embryo-fetal development and early postnatal development for biopharmaceuticals that are only pharmacologically active in primates, as described in ICH S6. However, there are additional considerations that limit the utility of studies in non-human primates for assessing some endpoints for DART risk assessment. See Annex 1 and ICH S6. Animal models of disease, genetically modified models and surrogate molecules can be valuable for investigating the effect of the intended pharmacology on development and reproduction. Studies in disease models can be of value in cases where the data obtained from healthy animals could be misleading or otherwise not apply to the disease conditions in the clinical setting. The model should be pharmacologically relevant and appropriate for the development and reproductive endpoints being assessed. The pathophysiology of the disease course in the model should be characterized. Some differences from the human pathophysiology would not preclude its use if these are unlikely to confound data interpretation. Animal-to-animal variability should be characterized and appropriate within the context of the study. If historical control information is limited, reference data for the study endpoints should be available or should be generated during the study to aid data interpretation. Genetically modified models can be used to provide information about on-target effects of a pharmaceutical on DART parameters through permanent or conditional alterations in target activity. Such models can inform on whether the biology of the target is closely linked to adverse effects on reproduction and development in routine test species. When the pharmaceutical does not have adequate activity against the target in the routine test species, surrogate molecules can be used to assess potential adverse effects on reproduction and development. Dose level selection, route of administration and schedule. The choice of dose levels, schedule and route of administration are important study design considerations and should be based on all available information, e.g. pharmacology, repeated dose toxicity, pharmacokinetics, and dose range finding studies. Guidance on the principles of dose selection for small molecules and biopharmaceuticals is given in ICH M3 and ICH S6. When sufficient information on tolerability in the test system is not available, dose range finding studies are advisable. Dose selection. There are a number of dose selection endpoints that can be used for DART studies. All endpoints discussed in this section are considered equally appropriate in terms of study design. The high dose in the definitive studies should be one that is predicted to comply with one or more of the concepts set forth in the following sections. The selected doses should take into account observations made in previous studies, and there can be instances where fewer than 3 dose levels are sufficient to provide the necessary information for risk assessment. Justification for high dose selection using endpoints other than those discussed below can be made on a case-by-case basis. Toxicity-based endpoint. This endpoint is based on inducing a minimal level of toxicity in the parental animals at the high dose. Factors limiting the high dose determined from previously conducted studies could include, but are not limited to, alterations in body weight, gain or absolute, either reductions or increases. Minor transient changes in body weight gain or body weight are not appropriate for dose selection. When assessing weight change effects, the entire dosing duration of the study should be considered. Exaggerated pharmacological responses, e.g. excessive sedation or hypoglycemia. Toxicological responses, e.g. convulsions, excessive embryo-fetal lethality, clinical pathology perturbations. Specific target organ toxicity that would interfere with the study endpoints within the duration of the planned DART study. Saturation of systemic exposure endpoint. High dose selection based on saturation of systemic exposure measured by systemic availability of pharmaceutical related substances can be appropriate. There is little value in increasing the administered dose if it does not result in increased plasma concentration of parent or metabolites. Exposure margin based endpoint. It can be appropriate to select doses based on predicted exposure margins relative to the exposure at the MRHD. For small molecules, a systemic exposure representing a large multiple of the human AUC or Cmax at the MRHD can be an appropriate endpoint for high dose selection. Doses providing an exposure in pregnant animals above 25-fold the exposure at the MRHD are generally considered appropriate as the maximum dose for DART studies. The 25-fold exposure margin should be based on a GLP-compliant dose range finding, PEFD or definitive study. Usually this multiple should be determined based on parent drug levels. However, considerations should also be given to ensuring an adequate exposure margin to major human metabolites. In the case of prodrugs, it can be more appropriate to establish the exposure multiple on the basis of the active metabolite, particularly if the test species has a lower ratio of active metabolite to prodrug compared to humans. The basis for the moiety used for the comparison, parent drug or metabolite, should be justified. For pharmaceuticals that have demonstrated PD activity in the test species only at exposures above 25-fold that are projected at the MRHD, higher doses can be warranted to assess adverse effects of exaggerated pharmacology. However, irrelevant off-target effects are more likely to be observed. When exposure-based endpoints are used as the basis for selection of the dose levels for EFD studies, TGA data from pregnant animals in a GLP-compliant study is expected. The choice for the use of total versus fraction unbound pharmaceutical exposures should be justified and consistent with the entire non-clinical development program as outlined in ICH S3A. Exposure-based approach for biopharmaceuticals. Exposure-based margins can be appropriate to select doses for biopharmaceuticals as per ICH S6. Generally, the dose should provide the maximum intended pharmacological effect in the preclinical species or provide an approximately tenfold exposure multiple over the maximum exposure to be achieved in the clinic, whichever is higher. ICH S6 should be consulted with regard to dose adjustment for differences in target binding affinity and other relevant factors. Maximum feasible dose endpoint, MFD. The MFD can be used for high dose selection when the physicochemical properties of the pharmaceutical or formulation associated with the route, frequency of administration and the anatomical, physiological attributes of the test species limit the amount of the pharmaceutical that can be administered. Use of the MFD should maximize exposure in the test species rather than maximize the administered dose. Note that changes to the frequency of dose administration can be considered to increase the total feasible daily exposure. Limit dose endpoint. A limit dose of 1 gram per kilogram per day can generally be applied when other dose selection factors have not been attained with lower dose levels. Selection of lower dose levels. It is generally desirable to establish a no observed adverse effect level for DART. The selection of lower dose levels should take into account exposure, pharmacology and toxicity, such that the dose response of findings can be established when appropriate. The low dose should generally provide a low multiple, e.g. 1 to 5-fold, of the human exposure at the MRHD. Those levels that yield exposures that are subtherapeutic in humans should be justified. Route. In general, the route of administration should be the clinical route. If, however, sufficient exposure cannot be achieved using the clinical route or the clinical route is not feasible, a different route should be considered. When multiple routes of administration are being evaluated in humans, a single route in the test species can be adequate, provided that sufficient systemic exposure is achieved, compared to that of all clinical routes, and that there is adequate coverage for the metabolites. Schedule. Dosing schedules used in the toxicity studies determine the exposure profile, which can be important in the risk assessment. Although mimicking the clinical schedule is often sufficient, a more or less frequent schedule can be appropriate. For example, twice daily dosing can be warranted with compounds that are quickly metabolized in the test species, although pragmatic factors such as study logistics, stress on animals should be considered when a more frequent schedule is contemplated. It can also be important to alter the dosing schedule to ensure that adequate exposure is obtained at all critical stages of reproduction and/or development being evaluated in a given study. This guideline covers vaccines, adjuvanted or not, used in both preventative and therapeutic indications against infectious diseases. While not within the scope of this guideline, the principles outlined can be applicable to non-clinical testing of vaccines for other indications as well. The types of reproductive endodevelopmental toxicity studies used for preventative and therapeutic vaccines depend on the target population for the vaccine and the relevant reproductive risk. Generally, DART studies are not warranted for vaccines being developed for neonates, pre-pubertal children or geriatric populations. For reproductive toxicity studies of vaccines, is it typically sufficient to assess a single dose level capable of eliciting an immune response in the man-animal model, using the clinical route of administration. This single dose level should be the maximum human dose without correcting for body weight. If it is not feasible to administer the maximum human dose to the animal because of a limitation in total volume that can be administered, or because of dose-limiting toxicity, whether local or systemic, a dose that exceeds the human dose on a milligram per kilogram basis can be used. To use a reduced dose, justification as to why a full human dose cannot be used in an animal model should be provided. The vaccination regimen should maximize maternal antibody titers and/or immune response throughout the embryonic, fetal and early postnatal periods. Timing and number of doses will depend on the onset and duration of the immune response of the particular vaccine. When developing vaccines to be given during pregnancy, a justification should be provided for the specific study design, based upon its intended use, e.g. protecting the mother during pregnancy or protecting the child early postnatally. Daily dosing regimens can lead to overexposure to the vaccine constituents. Episodic dosing of pregnant animals rather than daily dosing is recommended. Also, episodic dosing better approximates the proposed clinical immunization schedule for most preventive and therapeutic vaccines. Considering the short gestational period of routine animal species, it is generally recommended to administer a priming dose to the animal several days or weeks prior to mating in order to elicit peak immune response during the critical phase of pregnancy, in essence the period of organogenesis. The dosing regimen can be modified according to the intended vaccination schedule in humans. At least one dose should be administered during early organogenesis to evaluate potential direct embryotoxic effects of the components of the vaccine formulation and to maintain a high antibody response throughout the remainder of gestation. If embryo-fetal toxicity is observed, this can be further assessed using subgroups of animals that are dosed at certain time points. In cases where a vaccine includes a novel active constitutive ingredient, including novel adjuvants, consideration of additional testing strategies similar to those for non-vaccine products can be appropriate. Possible combination study designs in rodents. Although 3 separate study designs, in essence FEED, EFD and PPND, have been employed to develop the majority of pharmaceuticals, various combinations of these study designs can be conducted to reduce animal use. The main advantage of combination designs is that all relevant stages of the reproductive process can be assessed using fewer animals. Combination studies can also better mimic the exposure duration in the clinic, especially for drugs with long half-lives. A combination study design is a combined fertility and EFD study with a separate PPND study. Designs and study details for FED, EFD and PPND studies and combinations thereof can be found in Annex 1. In cases where no effects on male or female fertility are anticipated or where extending the dosing period is appropriate due to observation of reproductive organ toxicity in a repeated dose toxicity study, a combination design of repeated dose and fertility studies can be considered. After a defined dosing period within the repeated dose toxicity study, males can be paired with sexually mature females, whether untreated or dosed for at least 2 weeks prior to mating. This combination study can reduce the number of animals used, but the number of mating pairs per group should be at least 16. Further, if treated, dosing of females can be extended until the end of organogenesis, thereby allowing evaluation of EFD endpoints. Data reporting. Individual values should be tabulated in a clear, concise manner to account for all animals in the study. The data tables should allow ready tracking of individual animals and their conceptuses, from study initiation through study conclusion. Fetal morphologic abnormalities should be described using industry-harmonized terminology. All findings for each litter should be clearly listed by conceptus. Summary listings should be prepared by type of abnormality. The inclusion or exclusion of data from non-pregnant animals in summary tables should be clearly indicated. Interpretation of study data relies primarily on comparison with the concurrent control group. Historical control reference data can be used to assist data interpretation. Recent historical control data from the performing laboratory is preferable. contemporary data, typically from a 5-year period, is desirable and permits the identification of genetic drift. Statistics. Statistical testing to assess the significance of differences between the treated and control groups is expected in definitive studies. Many of the datasets from DART studies do not follow a normal distribution, necessitating the use of non-parametric statistical methods. Caesarean, fetal and postnatal data summary statistics should be calculated using the liter as the unit of analysis. Statistical significance need not convey a positive signal, nor lack of statistical significance impute absence of effect. Determination of biological plausibility based on all available pharmacologic and toxicologic data is often useful. Principles of risk assessment. All available data garnered from the pharmaceutical, related compounds, human genetics and knowledge of the role of target biology in human reproduction should be used to address potential reproductive risks in humans under the conditions of use, both during clinical trials and after marketing authorization. Any limitations, uncertainties and data gaps in the available non-clinical DART package should be addressed and their impact assessed. Generally, the results from definitive in vivo studies in an appropriate species with adequate exposures carry more weight than those from alternative assays or preliminary studies. Risk assessment is a continuous process through product development, as more information becomes available. Not all findings reported in DART studies are adverse. When a finding is deemed adverse, several factors should be considered in a weight of evidence evaluation for risk assessment. These can include exposure margins, biological plausibility, evidence of a dose-response relationship, potential for reversibility, the potential for confounding parental toxicity, and evidence for cross-species concordance. For rare malformations, the absence of increased frequency with dose does not always alleviate concern. Comparison of pharmaceutical exposure at the NOAEL in the test species to the exposure at the MRHD is an important component of the risk assessment. This comparison should be based on the most relevant metric, AUC, Cmax, Cmin, body surface area, adjusted dose. In general, there is increased concern when the NOAEL occurs at exposures less than tenfold the human exposure at the MRHD. Above this threshold, the concern is reduced. Effects that are limited to occurrence at more than 25-fold the human exposure at the MRHD are usually of minor concern for the clinical use of the pharmaceutical. The most relevant margin is generally the exposure metric in the most sensitive species, unless appropriately justified otherwise. Understanding the potential for reversibility will alter the risk assessment. Effects on male and female fertility that are reversible after cessation of treatment are of less concern. Conversely, critical irreversible developmental endpoints such as death or malformations are of increased concern. Embryo-fetal toxicity observed in the presence of maternal toxicity should be considered carefully to determine the likelihood that the finding is relevant for humans. Principal advantages and disadvantages to the use of various species utilized in DART studies are outlined in Annex 1. In vivo study design considerations. Generally, within and between reproductive studies, animals should be of comparable age, weight, and parity at the start. The easiest way to fulfill these factors is to use animals that are young, sexually mature adults at the time of the start of dosing. The number of animals per group specified in individual studies is a balance based on scientific judgment from many years of experience with these study designs and ethical considerations on the appropriate use of animals. Smaller group sizes can be sufficient to demonstrate anticipated adverse effects on reproduction or development at clinically relevant exposures of the pharmaceutical. Evaluation of 16 to 20 liters for rodents and rabbits provides a degree of consistency among studies. Below 16 liters, inter-study results become inconsistent, and above 20 to 24 liters per group, consistency and precision is not greatly enhanced. These numbers refer to liters available for evaluation. If groups are subdivided for different evaluations, the number of animals starting the study should be adjusted accordingly. Fertility and early embryonic development study. The FED study is designed to assess the maturation of gametes, mating behavior, fertility, pre-implantation development of the embryo, and implantation. The EFD toxicity study is designed to assess maternal toxicity relative to that in non-pregnant females and to evaluate potential effects on embryo-fetal survival, intrauterine growth, and morphological development. The PP&D toxicity study is designed to assess enhanced toxicity relative to that in non-pregnant females, pre- and postnatal, viability of offspring, altered growth and development, and functional deficits in offspring, including sexual maturation, reproductive capacity at maturity, sensory functions, motor activity, and learning and memory. EPP&D toxicity study is a study in non-human primates that combines the endpoints from both the EFD and PP&D studies. In this study, dosing is extended throughout the gestation period to parturition. Combination studies. The possibility also exists to combine study types to meet the goals of the development program. FED and EFD. The aim of the combined FED and EFD study is to test for toxic effects resulting from treatment before mating, males, females, through mating, implantation, and until the end of organogenesis. This comprises evaluation of stages A through D of the reproductive process. This study design is most often used with rodents, although it could be used with non-rodents. Male fertility and repeated dose toxicology study. It is also possible to evaluate male fertility during a rodent repeated dose toxicity study. In this combination study, males that have been dosed for a defined number of weeks are paired with untreated females. Following cohabitation, the males continue to be dosed until the scheduled termination of the repeated dose toxicity study. Female fertility and other FEED endpoints will need to be evaluated in a separate study. Data generated from qualified alternative assays conducted alone or in conjunction with one or more in vivo studies can be utilized to support hazard identification and risk assessment under limited circumstances. Potential uses can include circumstances where there is evidence suggesting an adverse effect on EFD, e.g. a mechanism of action affecting fundamental pathways in developmental biology, phenotypic data from genetically modified animals, class effects. Toxicity in animals species precludes attaining systemic exposures relevant to the human exposures under conditions of use, as support for a weight of evidence assessment when there are equivocal findings in animal studies, as partial support for clinical trials, including up to 150 WOCBP for up to 3 months duration, and pharmaceuticals being developed for certain severely debilitating or life-threatening diseases or late-life onset diseases. When alternative assays are used to support risk assessment, incorporation of these assays into an integrated testing strategy should be justified. Essays used for risk assessment should be conducted in accordance with GLP and qualified for context of use. In essence, applicability domain and regulatory conditions under which essay results are reliable. Strategies incorporating alternative assays should also assess the effects of drug metabolites when warranted. This annex does not recommend specific assays. Instead, basic scientific principles are included to assist in assay qualification for regulatory use. Alternative assays used to explore mechanism of action or otherwise not intended to substitute for in vivo derived EFD endpoints are not expected to be qualified in this rigorous manner. Qualification of alternative assays for prediction of methyl. Test methods must be appropriate in order for test results to be of value. Accordingly, the endpoints measured should be scientifically justified with respect to assay objectives and predictions. The relationships among the assays, predictions, endpoints assessed and the applicability domain should be supported empirically. To qualify an alternative assay or a combination of assays for use in risk assessment for regulatory purposes, a comprehensive description of the methodology and findings should be provided, including the following: a thorough description and justification of the predictive model, including which species and endpoints it is predicting. The currently available in vitro alternative assays used for evaluating potential hazards to development are designed to detect methyl. An evaluation of the biological plausibility of the model, including a description of the mechanism of embryo-fetal development, e.g. cell migration, differentiation, vasculogenesis, neurulation, gastrulation, and subsequent developmental adverse effects studied with the model. In addition, any limitations of each of the individual assays should be discussed. The description should include a discussion and supporting data to show that the duration and timing of exposure supports the prediction of methyl in vivo. An assessment of the accuracy and ability for the alternative assay to detect methyl. The performance of the assay is compared to the data generated from in vivo studies with compounds that induce methyl in the absence of confounding maternal toxicity. If the compound is not a marketed pharmaceutical, then in vivo data should be provided. A discussion determining whether an effect is negative or positive in the assay. Definition and justification of the threshold for molecular and metabolic markers predicting methyl. The details of the algorithm employed for determining positive and negative outcomes in vivo. The predictive model should correlate concentrations tested in the alternative assay to the in vivo exposure, preferably in pregnant animals, that results in an adverse outcome in the species being predicted. The list of compounds in each of the training sets, data used to discover potentially predictive relationships, and test sets, data used to assess the strength and utility of a predictive relationship, for qualification of the assay and the basis for the selection of these compounds. Data sources, e.g. literature, study reports, regulatory reviews, for all in vivo exposure and methyl data used for compounds in the qualification dataset, if not obtained from the reference compound list. Data demonstrating the test method's performance, covering an appropriate range of biological and chemical domains that are justified for the intended use of the alternative assay. Data demonstrating the sensitivity, specificity, positive and negative predictive values, and reproducibility of an assay or battery of assays to predict in vivo developmental outcomes. The performance of the training and test sets can be evaluated separately and or together provided the selected approach is justified. In cases when more than one assay is conducted, a separate description of the performance of each assay in addition to the integrated assessment used for the predictive model. A clear description of how the results of individual assays are integrated into the final prediction. historical data for assay development and use, e.g. viability, numbers and types of malformations, including positive controls. Examples of EFD testing strategies utilizing alternative assays. This section provides illustrative examples of integrated testing strategies into which alternative assays are incorporated to test for adverse effects on EFD. Potential approaches to defer in vivo testing as part of an integrated testing strategy can be found in section 4.2.3 of the guidance and slide 24 of this presentation. Pharmaceuticals expected to be embryo-fetal toxicants. For pharmaceuticals that are expected to adversely affect embryo-fetal development based on mechanism of action, pharmacologic class or target biology, it can be appropriate to confirm this activity in a qualified alternative assay. When a qualified alternative assay clearly predicts methyl at clinically relevant extrapolated exposures, this can be sufficient to identify the compound as an EFD risk, and further testing would generally not be warranted. If the alternative assay does not predict methyl, this should be confirmed in the definitive in vivo EFD studies in 2 species. Conducting these studies in series, as shown in this figure, can allow for reduction in animal use, as the second in vivo assay would not be warranted if the first one is positive. Under this scenario, since the pharmaceutical is expected to adversely affect embryo-fetal development, there is no merit in using in vivo EFD studies to attempt to negate a positive alternative assay response. Considering the risk-benefit for pharmaceuticals intended to treat severely debilitating or life-threatening conditions compared to less severe chronic diseases, where the likelihood of pregnancy is low, the use of qualified alternative assays can be considered an appropriate component of EFD risk assessment. When a qualified alternative assay clearly predicts methyl in the first species at clinically relevant extrapolated exposures, this can be considered on a case-by-case basis to sufficiently characterize the EFD risk. However, if the results are equivocal or thought to represent a false positive, definitive in vivo studies in 1 or 2 species should be conducted to assist human risk assessment. If no EFD signal is observed in the 2 definitive in vivo studies, at appropriate exposure margins, the results of the alternative assay could be considered of minimal concern for human risk assessment. However, for alternative assays that have been qualified to predict human methyl, additional data should be provided to support the conclusion that the alternative assay results represent a false positive finding. If one or both of the in vivo studies are positive for EFD toxicity, the compound is considered to be positive for EFD risk. Conducting the studies in series can allow for reduction in animal use, as the second in vivo assay would not be warranted if the first one is positive. If the alternative assay for the first species predicts a negative outcome, no methyl, a definitive in vivo EFD study in the second species should be conducted to confirm the assessment. If positive, the compound is considered positive for EFD risk. If negative, the compound is considered negative for EFD risk, and no further testing is generally warranted unless it is judged that additional studies would significantly alter the risk assessment. Pharmaceuticals In summary, the third revision of the ICH-S 5 guideline provides a focus on application to human risk assessment, offers additional dose selection endpoints, emphasizes the use of existing data, describes integrated testing strategies for assessing DART, including for biologics, and it outlines guidance on qualification of alternative assays for use in risk assessment for regulatory purposes. Following considerations are of importance. In the interest of 3 Rs, the experimental strategy should minimize the use of animals while not diminishing the overall human risk assessment. The revised guideline should be read in conjunction with ICH M3, ICH M3 Q&A, ICH S6 as well as ICH S9. With the implementation of Annex 1, in vivo study designs, and 2, alternative assays, of the guideline, it is anticipated that industry will generate and submit more results from combined in vivo reproductive toxicity studies as well as outcomes from qualified alternative assays. This presents a driver for increasing regulatory experience on their applicability, and hence the potential for harmonization will increase. Following conclusions are key for the guideline. For designing DART studies, all available pharmacological, toxicokinetic and toxicological data for the pharmaceutical should be considered in determining which study designs should be used. ICH S5R3 strengthens the role of 3 Rs, and it promotes novel testing paradigms for combined in vivo reproductive toxicity studies and alternative assays. This ends this presentation, but also presents the chance to start a new era of advancing established and implementing novel testing paradigms in developmental and reproductive toxicity testing to enhance human risk assessment while also potentially reducing animal use in a globally harmonized way.